Rattlesnake Venom Lab by Chloe Rodriguez and Jacob Jordan. Research question. How does temperature affect the activity of rattlesnake venom as an enzyme to deactivate its target? Background information. Macromolecules. Macromolecules are large, complex molecules that make up all living organisms. There is a total of 4 macromolecules, and there are carbohydrates, lipids, nucleic acids, and proteins. Enzymes. Each enzyme has an active site where the substrate binds and the reaction can occur. The different structures are primary structure, secondary structure, tertiary structure, and complex structure. Enzymes are biological catalysts that speed up chemical reactions. They lower the amount of activation energy needed for reactions to take place. However, temperature plays a key factor. At low temperatures, enzyme molecules move slower, resulting in enzyme activity being reduced. At high temperatures, enzymes move faster, resulting in an increase in activity. The temperature cannot go too high or too low because it will cause denaturation. In this experiment, we are simulating the effects of rattlesnake venom, which contains enzymes that break down certain molecules. We will examine how different temperatures will affect enzyme activity and time how long it takes to deactivate its target under these conditions. Hypothesis. If the temperature increases, then the time for the rattlesnake venom to deactivate its target will decrease because higher temperatures typically speed up enzyme reactions. However, at extremely high or low temperatures, the enzyme may lose its effectiveness due to denaturation. Our control group was room temperature. Experimental group was 40 degrees Celsius temperature water. Control group was room temperature water. And our controlled variables was 5 milliliters of water, 0.2 grams of sugar, 0.2 grams of enzymes, 5 minutes in designated temperature, and Benedict solution. Our independent variable was different water temperatures. The dependent variable was enzyme activity rate. Conclusion. Based on the experiment, the time for the enzyme to deactivate the target did differ between the cold and warm temperatures. For room temperature water, it took approximately 151 seconds, while 104 degrees water took 111 seconds. This showed that hotter temperatures deactivate the target faster, which supported my hypothesis. Some things that may have affected our results could have been measurements since the scale was hard to operate and time. If I repeated this experiment, I would do everything that I have now, but I would add a cold water bath to the experiment to determine how long it would take to deactivate the target.
The difference between gram-negative and gram-positive is that gram-positive has the outer peptidoglycan layer outside, while gram-negative has the inside two membranes that help to protect that lichen layer, because that is very important for the cell. And how do antibiotics work? This isn't that common of a thing that a bunch of people know, but I think it's important. So here's our antibodies. We're using this to just show. And here's our bacteria cell, okay? So what the antibodies do is they go into the lipid bilayer membrane, and what they do is they break through the first membrane. They get to these special enzymes that are on in the peptidoglycan that combine the peptidoglycans together to make the wall. So what they like penicillin, what they do is they cause them not to be able to bind, what causes the cell wall to explode and the whole cell to explode. So what this is showing is what happens when it doesn't work correctly. So the enzyme comes through like normal, and the protein goes in, attaches to the enzyme, and you can see those are still connecting. They're still connecting even though there's antibody attached to the active site. That's a mutation over time that bacteria like MRSA have developed. What this bacteria does is it can spread the bacteria through the horizontal gene transfer, which just helps it spread even bigger. Our medical intervention is to stop the spread, the horizontal spread. So what it does is our drug, that little dot, it goes into a bacteria cell. It goes through a protein channel in MRSA called PBP2A protein channel. It goes through there into the main area of the cell. There's no nucleus because it's bacteria, they don't have nucleuses. What it does is it takes out a small section of DNA and causes the cell to decease. This is very important because if we don't stop the immunity to these antibiotics, we're just going to keep doing them, and many more people are going to die. we need to stop overusing them so these special bug bacteria can't exist.